In the second YouTube, I will continue the procedure to explain the procedure how from calcium time series we can reconstruct astrocyte functional connectivity and the associated graph from a series of data acquired, and I will explain also a part of this which contain this image processing. So we'll directly move now to the calcium time series and signal processing. This is a study that we recently put on bioRxiv and hopefully will appear soon. And the result is a code that was written by Luzonka when she was a PhD student in the lab. And this results in Astronet, which is the name of the associated software. All right, so we are interested, so you can see in this movie, calcium activity in astrocytes, where you see all of these events here occurring spontaneously that are not predictable. And so what can we say about the causal relation? For example, if you think about this one and that one being activated later, can it be that it reveals some kind of underneath path or interaction that are more frequent? For example, if you think about this one compared to that one. And to do this, the idea is that maybe there is some underlying structure of interest. And that's what we are interested in here. So specifically, suppose you have a certain number of astrocytes. So just to explain that calcium is usually generated by IP3 receptors that are open with a mix of IP3 and calcium that leads to this calcium burst in a given astrocyte, and the calcium can go through this protrusion to activate through the gap junctions a neighboring astrocyte. And so if we look at this, it's not clear that there is a professional pathway. But so what we are going to be interested in is that the possibility that there is a path, that is, this one will first activate that one and this one and that one and so on, but not this one and that one first. There is no reason to expect that there is some preferential path, but so that was the idea of the study, to look at this more specifically. Okay, and so for this, you can acquire your data sets looking at slices and spontaneous activity. And the first part is to use some adaptive threshold image pre-processing to identify region where the fluorescence is going to vary, and it's not necessarily homogeneous. And so, but if you sum the movie, you can use this actually to identify specifically the astrocyte themselves or the region of interest. But then once the region of interest have been identified, you can go back to this region and look at the movie again and specifically identify, because you know now the border of your astrocyte, you can identify specifically how the signal will fluctuate in time. Then there is an event that I will discuss that you can smooth the signal to extract this little peak, although you can see here there's possibly a drift in the signal itself. But then what is interesting is that you can add a segmentation procedure based on local minimum to identify subregion, where in this subregion, and this will explain later, then we can define an order of activation of these different astrocytes, And by summing this event, that is each time two astrocytes have been activated, you look at this link here and you add a +1 if it was activated, resulting in what is called a graph, where you add this age between two astrocytes if they were sufficiently connected, and you can then threshold, saying that, for example, if you are suspicious that some were connected by random, at least you can say, well, I'm interested in only those that have been correlated or activated more than, let's say, 10 times. So you make sure that this was something that really happened. Okay, so now let's go back to the picture where you have identified the region where you have the astrocytes. Then you have the signal where you can identify the local peaks here, and you have to be careful because sometimes there are multiple peaks. So you have to use a procedure that allows to say when two peaks are two, they are connected, or they are in proximity, they are too close in time, you just take one of them. And then what is also interesting, once you have the maximum peak, you can accept that because you know that the time scale is few seconds, that there is some time scale before and after where you are going to let's say cut your signal, so that you can see here in the red line, and this is explained in details in the article, so that you have here a sort of moving lower envelope. You can also take actually the envelope for the empirical mode decomposition for those who are familiar, and then what you can do is use a spline just to smooth this. And once you have this, you just subtract the lower envelope to the signal itself. So this allows you to have no drift anymore, and you can see clearly the peaks. And then from the peaks, you can again go back to look at the number of peaks that you have here, or look simply at the maximum peak, and you can use this as a characteristic of one event at a given location. So this is illustrated here. You can see here that there is, as a function of time, a drift in the fluorescence intensity. So you can estimate here the value of the peak. You can get this lower envelope and this regularization procedure, and then you subtract so that you get these peaks in the end. All right, once you have this, what you can do is to look at the specific region of interest where you suspect that you have an isolated astrocyte, and then you look at the time evolution of this specific region that here are segmented. If you take here the minimum, so you have a certain number of subperiods that can be used to look at in this subperiod which of the astrocyte, let's say after the orange one, was activated in a neighborhood. So this is illustrated here. You have the first one which is activated, and then suddenly you look in your ensemble of astrocytes in your region of interest, the second one that was having a peak, and you choose the second one, and then you just create an arrow between the first and the second, and you can iterate the procedure so that you can go from the second to the third to the fourth, and so on, and in the end you get a path. And so we think that, and I will come back to this, that the notion of path is probably more accurate than just spiking or bursting activity as a proxy of a network, of the network activity. So this is an example of this segmentation of a few minutes of recording, where you have here the different paths, of a different period, and a different period you can calculate the path. So this is during event 1, event 2, event 3, and so on. So you get a certain number of paths, and from this path in the end, when you add each time two given astrocytes or two regions of interest were correlated in time, you observed that ultimately you can build a graph, and interestingly, there are subgraph which seems to be more connected compared to the overall graph that is present due to this reconstruction. And you can even take a threshold saying that I'm interested in, let's say, the subgraph which is built for connectivity that have at least, let's say, 5 or 7 or 9 and so on. So you want to make sure that the connection or the connectivity you see are sufficiently recurrent in your ensemble so that it means something. It's not just a spontaneous event. And so this is what we did here. For example, you fix a certain threshold. So let's say below 9 you don't consider, and you see what happened after 9, and this leads to this type of reconstruction. So now we've been interested in doing so in two type of regions. One is the hippocampus, which is a deeper region in the brain, which is segmented here with a classical view, and we have been interested in the CA1 region. And we did the same for the cortex here, which are shown here. There are several different types of cortex: premotor cortex, prefrontal cortex, and which is a collection of neurons organized in layers. So we said, are they fundamental difference in the connectivity of astrocytes in these two regions? And by applying the procedure, this is what we did. We found that by applying this with the same type of protocol, same duration of recordings, same preparation of the slides to make sure that they had the same type of spontaneous activity, we look at the cortex and the CA1 region and built the connectivity matrix. And when we run the procedure, when we increase the threshold of accepting a certain number of astrocytes where the connectivity had reached a certain value, when you increase this value and in the end you'll get almost no astrocytes, we could see clear difference between the two regions. So as you can see here in CA1 region, there was a persistence in the proportion of connected astrocytes, suggesting and showing actually that the connectivity was higher along this procedure in CA1 versus the cortex. And I think this is the first one that the first time that this result is shown here with respect to the present analysis. So this was not clearly known before, that we could have such difference in the network. And I would like to just make a parenthesis to illustrate the classical notion where in neurons we are looking at the coding rates in a population, where when we see a change in the coding, you see synchrony, you just simply look at the amount of spike that you have in your population. While here we think that actually that could be true both for neuron astrocytes, the path, that is the succession of astrocytes or neurons that are activated, is probably more accurate to describe how information is really moving and processed in these networks. So one thing also was important is to look at the passive diffusion of a dye in the hippocampus versus the cortex, which show how a dye can simply move across gap junctions. And we saw here no particular differences between the number of coupled cells in the hippocampus versus the cortex. So this suggests actually that probably the graph that we are reconstructing go beyond the simple notion of gap junctions, and there is maybe some specific laws, specific rules that are allowing the difference that we see between these two regions. So this is probably something to be studied in the future. And finally, to reinforce our reconstruction, we did a time-lapse analysis. That is, you have at a certain time t1, you acquire your data, you reconstruct your graph, you have your connectivity matrix, and 5 minutes later, under the same condition, you acquire a second data set with the same amount of time, and you wish that the connectivity matrix and the reconstruction graph, if your procedure is stable, would be similar. And this is actually what we've shown, showing that the procedure that we presented here is stable over a time-lapse analysis. Okay, so this is the quantification. There was no much changes after this time from time t1 to time t2. I'm not going to explain much of this. I think it's well explained in the paper, but the main idea is that this time-lapse analysis reinforce the methods. All right, so then you can, with this, look at statistics of this functional connectivity. You can look, for example, at the knot degree, or the number of hub astrocytes. The knot degree is the number of astrocytes that are connected to a given one, and the hub are the ones that are the most connected. And so from the statistics, you can look at the difference between the cortex versus the hippocampus, let's say here for the degree, or for the hub here. And here we looked also at the distribution of the weight. And we could see some differences between, again, those two regions. The sparsity measures the number of zero that you have in the matrix. The pass steps or lengths is a measure of how long are each of the paths in these two graphs. And again, we saw some differences that we quantify here, just using the p-value of the distribution, or here using a fit with a parallel function. So these connectivity matrices revealed that, again, there was difference in the connectivity. We could identify in the region of interest where are the most connected region, and I think this could be further explored in the future to see why exactly we have more connected astrocytes, although the underlying gap junctions and diffusion in the gap junction might not reveal much of the difference between the two. So that probably is something here to be understood. So the conclusion is that we've shown this time correlation and path reconstruction, which play a central role here in this analysis rather than spiking. The method allows to identify parameters of interest, such as the astrocytes. We here use this functional connectivity approach to distinguish, the difference between two regions, but there are probably much more to be done across the cortex and across other parts of the brain to explore the differences in the connectivity of astrocyte. And I think what also is important is to show some stability of this reconstruction through the time-lapse analysis. All right, I think I'll stop here, and we will go in the next YouTube about the notion of convergence. When you have many data sets, how much is enough to assess that you have reached some type of equilibrium and you're satisfied with the graph reconstruction. Thanks for your attention.